Okay, so we're looking at, as you said, read a study in the American Journal of Clinical Nutrition. This is a very important study. The who's who, all of the major players over the last good number of years, led by, of course, Dr. Richard Johnson and his team in University of Colorado, and David Perlmutter, who's on this as well, very well-known urologist and an individual who has written Brain Grain and a Drop Acid, which deals with uric acid in the body. This is an extremely important study. And what we're going to look at here is some of the findings. And what they talk about is that Alzheimer's disease may be a consequence of maladaptation to an evolutionary-based survival pathway, something that started millions of years ago. That was a survival pathway for mammals and for human beings that in modern life has become very injurious when it continues for extensive periods. And we'll explain that in just a moment. Now, this pathway can be engaged in multiple ways, including diets that are high in the two things we've talked about, fructose and its creation of uric acid. So let's jump into this. The first thing you need to know is what is the pathobiology of Alzheimer's and what are some of the stages? Well, there are 7 established stages of Alzheimer's disease, with stage 1 and 2 being considered as mild cognitive impairment. This is before real symptoms of Alzheimer's begin, and it's what we call basic forgetfulness. That can go on for quite a while. We put here 7 years, it could be 10 years, 15 years. It's quite a long period, which, by the way, is very important, because that is a period when there is the ability to reverse whatever is happening. You then continue into mild, moderate, and then severe Alzheimer's over a period of another 10 or 15 years, leading to the final stages with severe symptoms and the lack of physical control, etc. Clearly, one wants to deal with it before it gets anywhere close to that stage. And the transitions of the stages are based on, up until now, markers that are identified by PET imaging, that's positron emission tomography, and CSF assays, cerebral spinal fluid, which isn't easy to do. You got to go into the spine and get cerebral spinal fluid. So clearly, it is not an easy way of being able to detect what is going on. And what are you trying to detect? Well, up until today, we were looking at the 2 what are considered pathogenic plaques or accumulations of proteins, specifically amyloid beta and the tau protein, as well as other markers of neurodegeneration. And as I said, these are identified by cerebral spinal fluid, PET scans, MRIs, etc. We need a better way. But there is something that we need to rethink. And this has come out in recent months, in the last 1 or 2 years. And I'm quoting here from an article by Perlmutter, where we are looking at what the dominant theory of Alzheimer's disease has been and its treatment. And it's called the amyloid cascade hypothesis. Now, what does that mean? What it means is that they're saying Alzheimer's disease is caused by an accumulation of these amyloid beta proteins, these plaques, these accumulations of protein in the brain, and that triggers a chain reaction leading ultimately to neuronal damage, neuronal death in effect, and ultimately dementia manifested, in this case, by Alzheimer's. But there's a problem here, because multiple clinical trials of drugs that have been designed to reduce amyloid beta in the brain have failed to demonstrate significant clinical benefit. There's one that was just approved by the FDA. I can't even say it, aducanumab, go figure. These drugs have limited effect on improving cognitive decline. So yeah, they may be reducing the amyloid plaques, but they're not altering the progression of the disease. So there's got to be a rethink. The amyloid accumulation might be a symptom of the disease, not a cause. That's an extremely important difference. So really, when we look at the amyloid hypothesis, it's overlooking what seems to be really a complex multifactorial nature of Alzheimer's. In fact, Alzheimer's may involve other pathophysiological mechanisms. And what would they be? Neuroinflammation, oxidative stress. Remember this, we're going to go into it. Metabolic dysfunction. And particularly when we talk about metabolic dysfunction, we're talking about glucose metabolism and the way it starts to fall apart, and insulin resistance in the brain, which we'll go into in a few moments. So there's now growing evidence that Alzheimer's is in fact fundamentally a metabolic disease, which is deeply connected with lifestyle and environmental factors. Here we go again: diet, physical inactivity, chronic stress, sleep deprivation, to name just a few. So therefore, we need to have a more comprehensive strategy that considers multiple pathogenic factors that are involved in Alzheimer's disease. And that's what's warranted, to look at the multifactorial aspects, including interventions that are aimed at improving what? Metabolic health, reducing inflammation, and of course, addressing what in North America are disastrous lifestyle factors. So this busy slide, which I have discussed in a previous lecture, looks at the fructose survival pathway. And it's a busy slide, but an important slide. So I'll try to take you through it. And what it really deals with is that there is a reset in the cell, a reset of energy, which brings the energy to a lower level. Now we know that fructose has a standard caloric pathway. In other words, it can create energy, just like glucose does, which can be either stored as glycogen or some of the triglycerides, or it can be used immediately, just like glucose, to use for ATP, for energy in mitochondria. It can do that. But fructose is insidious because fructose has a uniquely separate pathway. And that pathway can be disastrous for the body because it's an energy depletion pathway. Now, what does that mean? Normally, in the mitochondria, we use ATP. ATP is used for energy. When it's used, it gets broken down into ADP, then AMP, etc. And normally, that flips back to ATP with a number of enzymes. They're called protein kinases. But in the case of fructose in an energy depletion pathway, that doesn't happen. What happens is that, number one, there's a rapid ATP consumption, but it doesn't get replenished. Why? Because AMP gets removed. It doesn't allow it to go back to ADP. It gets removed by another enzyme called AMP deaminase 2. And what does it form instead of ATP? It forms uric acid. And when uric acid forms anywhere in the body, but here we're talking about the brain, it starts to do several disastrous things. First of all, it causes mitochondrial oxidative stress because it reduces, here's number 3, it reduces ADP. And instead of going along the normal oxidative system in the mitochondria, it starts to stimulate glycolysis, which is much more laborious, doesn't produce the same amount of energy. You don't want that. Adding to that, uric acid inhibits AMP, protein kinase. Remember, I told you AMP would normally go back to ATP. Uric acid says, no, that's not going to happen. And it stops ATP from being reformed. So it reduces the ability of the mitochondria to get ATP recovered and to be used again. All of that causes inflammation and what you don't want, of course, and that's apoptosis, which is the death of the cell itself. All of that due to lower cell ATP levels. Why would that happen? Well, as I've mentioned in a big lecture I gave on uric acid, it's all part of what happened to the higher mammals and humans millions of years ago. It's a survival switch. It's what bears do in the winter when they're going to hibernate, what camels do when they need to preserve water. It's a survival switch. It's hunger and thirst begins to create the feeling of foraging. You go out and look for as much high carb as you can, glucose, fructose, whatever you can, because you want to create fat for the winter. You want to accumulate it. You create insulin resistance for the muscles because you don't want glucose to be used there. So there's insulin resistance, so the glucose can't get into the cells. You elevate the blood pressure and you cause local inflammation. All of that is fine if it's a survival switch, but we're not in a survival switch mode. So you need to look at uric acid's role in preclinical Alzheimer's. And remember, preclinical is important because that's the early stage when there's still a chance of reversing Alzheimer's. That's the key to this whole exercise. So what is the bioenergetics? Well, fructose, we know, raises uric acid levels and it lowers insulin signaling. So the brain cells, just like all cells in the body in this situation, lose their ability to use glucose. It can't get into the cell. They become insulin resistant. And there's an added feature, and that is the issue of nitric oxide. Now, we know a lot about nitric oxide. Well, what we do know is that increased uric acid levels create increased oxidative stress. Remember, oxidative stress is increases in free radicals in the cell, specifically in the mitochondria. And what that does is it decreases nitric oxide synthesis. We're not producing as much nitric oxide. Now, nitric oxide, we know, is a vasodilator. It opens up the blood vessels. If it can't, it compromises blood flow. Blood flow can't get to the brain as easily as it should. And nitric oxide synthase, which is an enzyme that makes nitric oxide in the brain, decreases, which makes it even worse. And that leads ultimately to reduced synaptic transmission, synapses, which allow cells to communicate. And if they can't communicate, you lose, amongst many things, memory. And how do we know that? There are a lot of studies on this. This is just one, looking at uric acid and cognitive impairment. If you take a look at the percentage of subjects who are scoring in the lowest quartiles, in other words, they're doing the worst in all of these aspects of cognitive function, general verbal, processing speed, working memory, verbal memory. And you look at how that happens given the amount of uric acid. Well, guess what? The more uric acid you have, the worse these individuals do. And here's the interesting thing. This graph is showing you what happens even when there are mild elevations in uric acid. In other words, these people are still in what's considered normal range of uric acid, and they're still having an increased risk of cognitive decline, especially in the elderly. So this is a real issue. And there are a whole number of other studies, and here is one that has come out in this latest article that we're talking about, and that is looking at what's called the polyol pathway, which I'll discuss in a moment, and how that activation affects Alzheimer's brains. When you look at fructose levels in the brains of Alzheimer victims, as well as sorbitol levels, those levels are 4 and 5 times the normal. Now, what does that mean? Well, the first thing you need to know is, what is the polyol pathway? Well, the polyol or sorbitol pathway is a 2-step metabolic pathway that does what? It converts glucose into fructose. And you need to remember that the endogenous production of fructose, meaning fructose produced in the body, can only occur when you convert glucose to sorbitol and then sorbitol into fructose. That's the polyol pathway. Well, as I said a moment ago, just imagine in people with Alzheimer's, there is a 4 to 5-fold higher level of both sorbitol and fructose when you look at the post-mortem brains. In this case, it was 9 patients with Alzheimer's compared to controls. This goes back about 7 or 8 years ago in a study that has been quoted here. So what does that tell us? It tells us that the hypothesis for the development of Alzheimer's disease needs to be reviewed. What are the risk factors in this hypothesis? Well, the risk factors are dietary fructose, a lot of sugar, which means glucose, sorbitol to fructose, and the real bugaboo, which is the high fructose corn syrup, which we have in almost everything in our diet today in the modern North American diet. It's a disaster. And with that, including also intermittent glycemia, those with diabetes, those with metabolic syndrome, those who are taking in high glycemic carbs, those who have a high salt diet, those who have had traumatic brain injuries, and those who have hyperuricemia, meaning they have high uric acid in the brain for any number of other issues in the body itself and the brain. What happens to them? Well, you cause fructose metabolism. And not only in the whole body, but specifically here, we're talking about the brain. Cerebral fructose metabolism, which I just showed you in another slide, goes to uric acid. What does that do? It causes neuroinflammation, which you do not want. Mitochondrial oxidative stress and loss. Not only is there oxidative stress, which is bad enough, but you start destroying the mitochondria. And that is disastrous for the cell. It leads to the cell's inability to survive, and it causes its own death, apoptosis. Remember, I told you that fructose and uric acid cause cerebral insulin resistance. Glucose can't get into the brain cells, and it causes impaired glycolysis, reduced ATP, which was part of that ancient survival pathway, which is now destroying us. The consequence of all of that is a reactive injury. It's a symptom of what's happened prior to that. It's what causes the amyloid and tau protein plaques and eventually neuronal deaths. So the various risk factors here can induce cerebral fructose metabolism that can lead to pathological and clinical manifestations of Alzheimer's disease. So what are the key takeaways? Chronic and persistent decrease in cerebral brain metabolism that is driven by what? By recurrent fructose metabolism, that intake of high fructose corn syrup, etc., leads to progressive brain atrophy and neuronal loss with all of the features of Alzheimer's disease. So the effects of fructose on the brain, which originally was supposed to be a survival mechanism to stimulate foraging, reduce cerebral energy demands, has now been amplified by the Western diet, and it has resulted in severe reduction in energy that is required to maintain the neuronal cell needs. And that leads to what? To neuronal injury and death.